The gas exchange system of insects, even though it's diffusion-based, is not entirely physical. The insect is not at the mercy of the constraints of fixed law, but its master, using physiology to do clever things within the physical bounds set by fixed law. Let's look at two of these, one pretty simple and the other a bit more complicated. The second one is instructive because it shows elegantly how insects have learned to play off the physical properties of the different respiratory gases against one another. Living things have to adjust fluxes to demands. In the context of the insect tracheae, this means adjusting physiological delivery of respiratory gases to the usage rates of local tissues. Insects do this by using water to adjust the diffusion properties along the path lengths for diffusion along the tracheals. Here's a simple diagram of a tissue. We're making it look like a muscle, with a branching network of tracheals serving it. Let's assume that the entire tracheal system is filled with air. This means that along the entire path length of the tracheals, movement of gas will be governed by the binary diffusion coefficient for oxygen in air. However, the ends of the tracheals are not always filled with air. They can, under the control of local tissues, fill partly with water. This means that there are now two diffusion paths in series that oxygen must follow to the tissues. Most of the path is still governed by the high diffusion coefficient for air, but when the tracheals are partly filled with water, diffusion along the terminal part of the path is now governed by the low diffusion coefficient for oxygen in water. By adjusting the degree to which the tracheals fill with water, the insect can adjust oxygen flow along the tracheals to match local oxygen demand. If the tissue is experiencing low oxygen demand, the tracheals are allowed to fill up with water, and oxygen diffusion to those tissues is slowed. But if the oxygen demands for the tissues goes up, the tracheals can be drained of water, opening up the entire pathway to the very high diffusion coefficient for air. That was simple. The next mechanism is a little more complicated, and it involves a phenomenon called discontinuous respiration. This is a phenomenon that is found most commonly in insects that are well adapted to arid conditions. We'll see that connection in a few minutes. If we put an insect into a respirometer where we can measure the molar fluxes of oxygen and carbon dioxide between the insect and air, we find that oxygen consumption ticks along at a more or less steady rate, but that carbon dioxide emission occurs in periodic bursts. These bursts are what is meant by discontinuous respiration. Okay, let's set that observation to the side for the moment and analyze more closely what's happening to the balance of respiratory gases in the tracheal. Let's suppose we have a tracheal feeding a cell. There will be a molar flux of oxygen from the tracheal into the mitochondrion, sufficient to meet the mitochondrion's metabolic demand for oxygen. At the same time, the mitochondrion will be producing carbon dioxide. Remember now that carbon dioxide in water immediately dissociates to bicarbonate and protons. The important point here is that there's a net molar flux of a gas, oxygen, out of the tracheal into the cell, but there's no replacement flux of carbon dioxide gas from the cell back into the tracheal. The carbon dioxide is stored in soluble bicarbonate. This imbalance makes the tracheal act like a tiny suction pump, drawing a mass flux of gas from the spiracles into the tracheals. This is known as a differential solubility pump, because it depends upon the differential solubility between oxygen and carbon dioxide. This is where that large differential in Bunsen solubility begins to matter. This gets us only partway to an explanation for discontinuous respiration, though. The rest of the explanation involves the spiracles. Here's a cross-section through a typical spiracle as it connects to a trachea. The spiracle itself, that is, the opening in the outside body wall, opens into a complex internal chamber which connects ultimately to the trachea. One part of this chamber is called the occluding chamber. The occluding chamber contains a valve which can open or close. Thus, the connection between the outside air and the trachea can alternate between an open state and a closed state. Returning now to discontinuous respiration, there's a clear correlation between the bursts of carbon dioxide emissions in the insect and the state of its spiracles. Specifically, during the CO2 bursts, the spiracles are in an open state. What happens in between the open states is a little more complicated. The spiracle can exist in a closed state, but it can also exist in a partly open state, called fluttering, where the occluding chamber opens and closes very rapidly. Immediately after the burst of carbon dioxide emission, the spiracle closes down tightly, but only for a short period. The remainder of the time, the spiracle is in a fluttering state until it opens fully again. Clearly, there's something complicated going on here. The easiest place to start clarifying things is to look at how the internal pressure inside the trachea changes during the closed part of the cycle. During the latter part of the fluttering phase and the open phase, the pressure inside the trachea is very close to atmospheric. When the spiracle closes, there follows a steady drop of pressure inside the trachea. When the spiracle begins to flutter, there's a gradual return of pressure back to being close to atmospheric, where it stays until the spiracle closes again. The explanation for this drop of pressure is pretty straightforward. The molar imbalance of respiratory gases at the tracheal, that is, the continuous removal of oxygen gas from the tracheal without it being replaced by carbon dioxide gas, produces a steady decline in the total quantity of gas contained within the tracheal system. Because the spiracle is closed, this reduction of gas volume cannot be offset by an influx of gas from the atmosphere. By closing its spiracles, the insect has literally put a cork in its tracheal system, and the result of the gas drawdown at the tracheal is a drop of pressure. The gradual return of tracheal pressure when fluttering begins is also pretty straightforward. The insect partially opens up its tracheal system to the atmosphere, and gas seeps in under the influence of the suction pressure inside the tracheae. We can get an even clearer picture of the events if we look at the partial pressures of carbon dioxide and oxygen inside the tracheae during this cycle. During the closed phase, the PO2 declines at a steady rate as respiration draws down the total quantity of oxygen in the tracheal system, and the PCO2 increases at a steady rate. During the open phase, there's an abrupt turnover of oxygen and CO2 within the tracheae. PO2 increases dramatically and PCO2 decreases. That's the explanation of the burst of CO2 during the open phase. During the fluttering phase, PO2 stays steady at a mole fraction percent of about 4%, whereas the PCO2 continues to increase gradually. The steady PO2 during the fluttering phase indicates that there's a steady inward flow of oxygen through the spiracles into the tracheae, borne along by the pressure-driven mass influx of air. The PO2 is steady because oxygen is being replaced by this inward flow as rapidly as it's consumed by the cells. The PCO2, of course, continues to increase because the carbon dioxide continues to accumulate in the tissues. That's not quite the end of the story, however. Let's remember again the basis of CO2's high solubility compared to oxygen, namely the fact that CO2 reacts with water to form carbonic acid, then protons, plus bicarbonate. As CO2 accumulates, the protons and bicarbonate are sequestered from one another, with bicarbonate accumulating outside the cells and protons accumulating inside the cells. As long as bicarbonate and protons are sequestered from one another in this way, it will be impossible for CO2 to leave the system as a gas. During the open phase of discontinuous respiration, however, protons are released from the cells, and this pushes the equilibrium of this reaction towards CO2, which enters into the gas phase in the tracheals and contributes to the turnover of CO2 gas in the tracheae. Well, that was awfully complicated, but it's worth it to go through in detail because it illustrates two important things about physical constraints in physiology. The first is the clever way that the tracheal system of insects plays differentials of solubility of the two gases, oxygen and carbon dioxide, against one another. By exploiting these differential solubilities, the insect produces a so-called differential solubility pump, actively sucking air into the tracheae. As a side note, this helps reduce water vapor loss from the tracheae because the inward flow of air impedes the outward flow of water vapor by diffusion. This may be one reason that discontinuous respiration is prevalent in arid-adapted insects. It conserves water. The second is how the insect can turn this whole thing over in an instant, simply by changing how it sequesters protons and bicarbonate. The insects are not constrained by fixed law, they are its masters. Okay, let's now turn from deserts to water for a final example of how insects can play differentials of solubility and diffusion against one another to enable them to inhabit environments where physiologically they should not be able to live.